Well, for our second talk, we have Dr. NATALIE Twine, who is the team lead for Genomics Insight at CSIRO. She's going to be talking today about a couple of very interesting projects in which she's combining the knowledge about bioinformatics and the tools that we are used to use for data science, Spark and machine learning, with the knowledge about genomics. And she's going to tell us about two very interesting applications. One is the project Tribes, and the other is about detection of diseases based on genomic data. So without further delay, please help me receive NATALIE to the stage.
Hi everyone, thanks for listening to me today. So changing tack slightly from decision making to DNA. I'm going to tell you a little bit about using machine learning and cloud computing to explore DNA and unlock the secrets in your DNA. Okay. So I work at the CSIRO. I head up the Genome Insights team for CSIRO, and today I'm going to talk to you today about two platforms that we've developed in our group. The first is called Variant Spark, which is focused on reading genomic information to identify disease genes in the billions of DNA molecules that we all have. And the second is on Tribes, which is about discovering ancestry, distant relatives through DNA sequence, which will help us to uncover disease genes. So at the CSIRO, I don't know if all of you are aware of the CSIRO. They're Australia's National Research Institute, a research agency, and probably most well known for the discovery or invention of Wi-Fi. Also, CSIRO have invented the Hendra virus vaccine. They've got the Hendra virus vaccine, as well as many popular diets in Australia, namely the total well-being and low-carb diets. And within CSIRO, I work within the eHealth Research Centre, and our vision is to improve healthcare through digital technology and services. So, most of you are in a corporate or business environment, and you may be asking the question, well, how is DNA research relevant to me? Well, a lot of the challenges that we're facing in the research that we do at CSIRO and within my team are, in fact, coming to your domain or already in your domain. And I'll talk a little bit more about that throughout the talk today. So first of all, just to get some bits of biological information out there, which you may or may not know. So hands up, who knows what a genome is? Okay, about half, a third. So the genome effectively holds the blueprint for every cell in our body. And the genome is made up of DNA. So what is DNA? So DNA is a molecule that's present in all of our cells, which is effectively a set of instructions which just defines how we are as humans. And the genome is then the collection of all the DNA molecules in a cell, which becomes the blueprint for that cell and how we are as individuals. So every person in the room has a unique genome, so it's effectively a fingerprint which defines you as unique. And then when we drill down and we look at the DNA within the genome, there's around 3 billion letters of DNA within a single genome, and this, as you can imagine, translates to a lot of data to analyze when we're looking at causes of disease and what's really going on at the DNA level. So our genome affects a lot of things in our life, from how we look. So I would say around 30% of the people in the audience, you can do this if you like, have the hitchhiker's thumb, which means you can bend your thumb all the way backwards. And the rest of you will have a thumb that doesn't bend very far backwards, a bit like mine. From what diseases you get, so your risk for getting cystic fibrosis, for example, is inherited. And then also your behavior. So whether or not you like coriander, again, based on the statistics, around 16% of you in the room should dislike coriander and the rest of you should like it. So I don't know if that bears up. We can do a score later. So as I mentioned earlier, alluded to earlier, because of these 3 billion DNA bases in our genome, the data size is astronomically large. And in fact, I'd argue that it's genomically large. Because if you were to stretch out all the DNA bases in your genome, which is normally tightly wound within a cell, it will stretch out to 2 meters. And if you can imagine a cell is microscopic, you can't see it without a microscope, that's a lot of DNA. And then if you multiply that by the number of cells in our body, which is 100 trillion, this is a huge amount of information that we have to analyze. It's in fact larger than the universe. So I'm going to start off my talk presenting the Variant Spark platform which we've developed, which is a machine learning approach to analyzing genetic variants. And doing so, we can identify disease genes within the genome. And the application case that I'm going to talk about today, for which we've developed, one of the uses for this platform is amyotrophic lateral sclerosis or motor neuron disease. So this is a devastating disease which leads to death within around 2 years, and you may know about it through the Ice Bucket Challenge, which was pretty popular on social media a couple of years ago. And through our we have access to very large data sets from genome sequence data for ALS patients. And this is through the International Project MIND organization, where we have access to around 15,000 ALS patients and around 7,500 healthy individuals as controls. And this is across Europe and Australia. So if you imagine, we want to identify within the ALS patients what within the genome is causing them to have disease, whereas healthy people don't have disease. And if you look at a very simplified example where we have just 2 genes in the genome, which is, in reality, you have around 23,000 genes in your genome, but just for example, so we have 8 individuals here, so every blue line represents an individual's genome, and we have 2 genes in this particular genome. And then if we can highlight within the genes or within the genome the positions that are variable, so there's positions along the genome which vary amongst each individual, and that's what contains the useful information, this variable information. And here we're looking for patterns which are different in the sick people versus the healthy people. And if we label the people, we label the genomes with their binary labels, so sick or healthy, case control, we can see quite easily in this toy example that that position there is what's distinguishing between healthy and sick. So as I said, this is a very simplified example and obviously not what happens in reality, but just to explain the principle. So a lot of diseases that are prevalent today, including ALS or motor neuron disease, so diabetes, heart disease, cancer, these are all what's known as complex disease. And complex disease means that they're driven by not one gene, not 2 genes, but multiple genes. And the variant positions within these genes interact to drive the disease or to trigger the disease. So to be able to identify these type of interactions and this type of information within the genome, we need a more sophisticated approach. And what we use is a machine learning approach, which is quite well known, random forest approach to identifying the genetic variants that are associated with disease, as well as the interactions between the variants. So when you translate that to an actual number in this data set, so we have around 22,000 individuals and 80 million features for each individual. So each genome has 80 million features, which is the variable positions. This translates to around 1.7 trillion data points, which is a lot of data. And what we built is a machine learning platform to identify the disease genes from this data set, and where we know the disease status. So we know the outcome, which is disease or healthy. But this methodology can be applied to other domains. It's not sort of restricted to biology. So the features could be not necessarily a genomic profile, but they could translate to time series data, concatenated data, sensor data, or log files. And the binary outcome, it doesn't have to be disease, it could be churn rate, occurrence of failure, product quality. And again, instead of hunting for disease genes, you could be looking for predictive markers for that, for a particular outcome. So, such large-scale genomic data sets, or population-scale genomic data, so we're looking at whole populations of people or populations of disease, really require big data solutions. So a standard compute solution isn't going to work for us, or even high-performance compute, which is quite popular in academic domain or bioinformatics field, is sort of restrictive in that it is bound by a particular number of nodes and it's more compute intensive. And so the solution that we use in the technology we developed is the Apache Spark platform or framework. And this is really useful for this context because it's data intensive and not bound by a particular number of nodes, and is also very parallelizable. So the platform we've developed is called Variant Spark, and when we benchmarked it against other random forest-based machine learning methods on the same data set, it performs really well in terms of speed and accuracy. So we can analyze around 3,000 individuals with 80 million features in just 30 minutes. So Variant Spark is available on all the major cloud providers as well as on-premise, so it's on GitHub for free. And we strongly believe within our group is about democratization of technology. So really enabling access to technology and making it easy to use. So to do this, we make use of all the major cloud platforms from AWS, Microsoft Azure, Google Cloud. And most recently, we've also just launched Variant Spark on AWS Marketplace, which is quite exciting. So it's available internationally. And that was actually in today's AFR, the fact that it's been launched on Marketplace. And this is actually the first publicly available tool for health data that's been launched on Marketplace. So that's really quite cool. So if anyone's interested in trying out Variant Spark, we do have some demo notebooks, so in a Jupyter Notebook environment on Databricks. So Databricks is a managed Spark cluster. Anyone can go and log in. You just head on to the URL on the screen. And we have the notebook set up with some publicly available genetic data from the 1000 Genome Project. You can upload your own genome data if you have it. And the little example we have is the hipster index. So this is a fake phenotype or fake physical outcome. Well, it's a real physical outcome, but we've sort of selected genetic variants that we think are important for being a hipster. And those are ability to see checks, so check patterns. Some people have a better ability than others. Caffeine metabolism, so how well you metabolize coffee. And also your hairiness. So all the genomes. So you can put your genome sequence data into this tool, into the Variant Spark. You can run the machine learning model on it, and it will output the features that are driving your genome to be hipstery or non-hipstery. So that's a little fun example as a demo. Right, so moving on from Variant Spark, the second part of my talk is more around ancestry and the platform that we've developed for detecting distant relatives. So you probably know that disease is often inherited, or your risk of disease. So for example, your risk of heart disease, cancer, diabetes is partially inherited. However, these inheritance patterns are quite complex, meaning that it's not just because your father had a disease doesn't necessarily mean you will. It might transfer through more, it might be sort of skip a few generations, or you'll need to inherit multiple genes to present with the disease. So the platform we built to identify distant relatives is called Tribes. And just to give you an idea of the amount of genome data, because to identify these distant relatives, we need genome sequence data. And just to give you an idea of the amount of genome data that's out there or predicted to be out there, by 2025 it's predicted that around 50% of the world's population will have been sequenced. And if you look at the current sources of big data generation, so which are astronomy, YouTube, and Twitter, by 2025 genomics will completely dwarf those data, sources of data generation. And this sort of explosion of genomic sequence data has really helps us in exploring genetic ancestry, because more and more people have their genome sequenced. And actually, one of the applications for genetic ancestry, which has been in the news quite a lot, is this identifying criminals. So the Golden State Killer was last year he was identified through a distant cousin that they used ancestry software to identify through a genealogy website, so through genome sequence data. So moving from finding criminals to fighting disease, again, back to the application case of ALS or motor neuron disease, which I've explained to you before. So as part of our big international project with Project Mind, so the international consortium where we have this huge cohort of ALS patients, our collaborators at Macquarie University have contributed 850 Australian whole genome sequence data. So 850 Australian patients with ALS have had their genome sequenced, and we have access to this data. And we wanted to ask the question, like, how many are there distant relatives within this cohort, and is that going to help us to drill down and really identify disease variants or disease genes that are involved in the disease? So you might ask, how do we identify genetic ancestry? So how do we identify if people are related to each other? So the sort of unit of information that's really key to, that sort of underpins this technology is really identifying matching segments of DNA that are shared between two individuals. And then also that segment is inherited by a common ancestor. So if we look at the example on the left, again, this toy example where we have 3 generations of first row being grandparents, then second row parents, and then last row children, we can see this shared segment that's highlighted in the 2 green arrows and coloured in orange has been inherited from their grandfather, whereby, so each shape represents an individual, where the squares are men, the circles are women, and at each generation you have 2 copies of your genome, one from your mum and one from your dad, and at each generation's children basically have a reshuffled genome, half from their mum and half from their dad. But this occurs sort of in a random process of reshuffling, so you'll get chunks of your genome from your dad and chunks of your genome from your mum. And by identifying these segments or these chunks of DNA that are shared between individuals, and then also that they've shared through a common ancestor, so in this case, the 2 cousins share an orange segment due to their grandfather, we can then tell how closely these individuals are related by seeing how many segments they share and the size of the segments. So these segments are known, the technical word for them is IVD segments, or inheritance by descent. And these segments can help us to sort of prioritize genetic variants involved in disease. So they can help us to zoom in on the regions of the genome that are involved in disease. So if we imagine the individuals with ALS, we identify those who are related to each other, we can see which segments of their genome they share, and then we can really focus in on those regions to help us to prioritize which genes to explore. Or, I guess in more computational terms, to narrow down the search space. So it can dramatically narrow down the search space by using this approach. So the platform we built is called Tribes. It's a pairwise relationship classifier. So if we put in the cohort of 850 patients, the genome sequence data will have a relationship for every pair, from no relation to first degree. And we've shown that it's substantially more accurate than gold standard tools. So if we have the plot on the right with the accuracy, where the the percent true positives is on the y-axis and the degree of relationship on the x-axis, from first degree through to seventh. So first degree is parent-child and siblings, second is grandparent, third is cousins, etc. And you can see, so tribes is in orange, it's outperforming the gold standard tool. The accuracy does drop off after third degree, and this is purely due to the fact that you share less and less of your genome as you get more distantly related, and there's a lot of randomness involved. So it's hard to get high accuracy as you do share, as the relationships get more distant. So again, we've applied our methodology to our cohort of ALS patients to identify novel relationships and disease genes. And one of the interesting things that we found, so within these 850 patients, we do have about 100 of them which did know, they knew that they had a family history of the disease. So they had relatives that have come into the clinic in Macquarie where we collect the data. So some of the families are small, say like 3 or 4 people, others were big, 10, 20. And what we found is, in fact, these families, these 25 families that had ALS, in fact, were related to each other distantly as sort of super families. So we've been able to identify, if you can imagine, each family is represented by a different color. So each circle represents a family and a different color. And using a sort of the output from tribes and then clustering all of the results, and we focused on a particular gene here that is known to be involved in ALS, SOD1. We have been able to identify these sort of distant relatives across 19 different families. So effectively what this means is at some point in evolution, a single individual has had a mutation in their gene which has caused the ALS, and then this is sort of perpetuated through their forebears or descendants, and all of these individuals who are clustered together are their descendants. So this is quite exciting, and we've just published this. And some other interesting things that we found in the data set is we actually, in addition to sort of clustering families into super families, is we've been able to identify 53 new relatives up to 7th degree. So 7th degree is your great-grandfather. And in doing this, as I mentioned before, so that the benefit of identifying distant relatives is you can really narrow the search space to identify which genes are important. So we narrowed the search space to around 2% of the genome for particular subsets of individuals, and we were able to identify 2 novel disease genes, which our collaborators in Macquarie University are currently testing in the lab in their animal models. So 3 things I'd just like you to take away from my talk today. First is that datafication in all industries, especially in genomics, requires not just big data solutions, but wide data solutions. So a lot of features in the type of data that we're looking at. And to deal with this, we've developed Variant Spark, which is publicly available. And also, discovering distant ancestry is more than just useful for curiosity purposes, but it actually can help to hone in on identified disease genes. And then finally, that sort of business and life science research are not that different. And at CSIRO, we always like to work with industry, and if you're interested in any of the technology I've talked about today, so please come and have a chat. Just thanks to the rest of the team and group, and Dennis, who heads up our group, and people at Data 61 that we work with. And then finally, check out our website, get in touch if you're interested.
Thanks, Natalie. I think this is a fabulous presentation. And I think it's a good showcase of what ML can do for good. But I think it's also a good example of the risks in terms of the ethical risk and data privacy. I just wonder if you also have anything to add to this from that angle.
Sure. I mean, yeah, that's also something that we're also concerned about and interested in. And one of our research projects, in fact, is on genomic privacy. So the fact that we're putting all our tools in the cloud means that the data does need to be very secure. And it's an ongoing research area because effectively we work in a collaborative way, so we often share data sets with other institutions or other organizations, but everyone wants to maintain the privacy of their data, and also data costs money, so they don't want to release it. So there are technologies that we're working on which enable sort of privacy data to be analyzed in a private way, but you still can get the sort of the model outcomes, the machine learning outcomes can still be used while maintaining that privacy.
You used ALS as a specific example.
Yes.
I gather because that was identified as a disease that was very genetically based. So you specifically mentioned that it was identified that the genetic mutation arose 5 times independently. That sort of thing certainly does happen. But it's,
that was just for one gene. Sorry, I didn't really preface the background to ALS. It's a very complex disease, and it's partially genetic, partially environmental. And a lot of patients, we don't know what's causing the disease. There's a small cohort which have that mutation, so it's like 10% of patients have this gene that we know is causing disease, but it's a very small proportion of all of the people with ALS. So it's not the same gene that causes ALS in different people.
so of those people that you worked with in Australia,
Yes.
I guess you would have not found any specific correlations in fair few, like 40% or something.
Sorry, can you say that again?
If there was a basis that was not genetic,
Oh,
then
yeah.
I guess of that population size that we used, a fair proportion of them would not have uncovered anything useful.
Yeah, I mean, this is partly why the consortium arose that I talked about, the Project Mind consortium, because it's kind of so complex and there's so many genes involved, the bigger the sample size, the better, as you know. So the more number, the higher your N, the bigger power you have. And so what we think, actually, a lot of the schools of thought now is that ALS is driven by rare genetic variants. So it's very small proportions of the population have a variant that are driving disease. And yeah, like I said, you having these huge consortiums of data really gives us the power to identify the fact that this rare variant is associated with the disease. But when you're looking at, 100 or 200, and it's at a frequency of 0.00%, you're going to get 2 which have them. So it's just not enough power. So that's kind of why it's important to collect as much data as possible, and then also why we need to develop these tools which can handle such big volumes of data.
Yeah, thanks. You mentioned having a binary classification. I was just wondering how many of the healthy patients maybe weren't showing any symptoms but did have a genetic susceptibility to the disease?
That's a very interesting question. So what we do with the controls, so ALS tends to onset around middle age, like 50 plus, from 50 onwards. And we have the controls are age matched. So there would only be 50 plus individuals because by that age, you would generally have presented with the disease. So again, it's a good question, and having sort of a control group which are 20-year-olds isn't useful because they may present in 30 years' time with the disease. It's a great question.
Hi. Over here.
Hi.
Hi. A couple of questions, both about some of the graphs that you did. Firstly, the accuracy rate of tribes. I was like that
True
was
positives. Yeah.
That was on you're using true positives for that.
Yeah.
I'd like to hear about the false negatives as well. So people that were obviously family that tribe sets were not.
Yeah. So there is some. I know I didn't plot. I didn't plot false negatives. People have asked me that question before. I should probably change the plot to include the false negatives. There are some false negatives. I can't put an exact number, but you can do sort of post hoc analysis to identify whether or not the result is. You can do sort of post hoc analysis to see if the relationship is real or not.
Right.
So what I mean by that is when I talked about the segments. So if you share a lot of really tiny segments with the supposed relative, it's a reduced chance that that's a real match. Whereas if you share one long segment and a few tiny segments, that's a much higher chance because the algorithm is basically predicting these segments, and it's easier for the algorithm to get it wrong when it's lots of little tiny segments. Yes. But it's harder to get it wrong when it's one really large segment.
One more question. If you just go back to the graph about the speed and accuracy of the
for variance spark.
Yeah, if you don't mind. There we go. I was just wondering what sort of measurement you're using for speed there, because usually the higher would mean text slower.
Sorry, what do you mean?
Like usually speed would be kind of like measured almost in seconds, you know, like the seconds it takes to.
Yeah. That I can't tell you because I didn't generate this plot.
Right,
so I can't tell you.
Don't know, like,
I don't know the units.
Like
I don't know what the units
kilobytes
are.
per second.
Yeah, I don't know what the units that
Right.
they used in this plot.
Okay,
Sorry.
right. Thank you.
But good question.
Hi, Natalie.
Hi.
It's Mark from 23 Strands. Thank you very much. It was a great talk.
Yeah.
I've got a question around Mark Zuckerberg and his recent comments around Amazon AWS,
Yes.
particularly from BioHub. What were your thoughts on that?
You're going to have to tell me what his comments are because I didn't hear those.
The AWS in the cloud is not turning out to be as cheap as they expected.
Okay. It depends. When you say cheap, what are you comparing it to? Because for us, the alternative is using high-performance compute system, which requires a lot of infrastructure, people to manage the infrastructure, infrastructure that sits idle a lot of the time. So when you're comparing it to that alternative, it's definitely cheaper. And also, it's just, again, compute on demand, so it enables a lot more people to use it. You don't have to have that infrastructure in place to be able to run the genome sequence data through the algorithm. So for us, that's the benefit. No? You're not happy with that answer?
Not really, no, sorry.
So what do you mean when you said it's not working out as cheap as it should be?
He was pretty critical, and I'm just out of...
Yeah, I haven't heard the speech, so...
We're doing a bit of whole genome sequencing as well.
Oh, okay.
As an example, the upload costs, the transfer costs, because they're big data files. And then I think even keeping FPGA platforms running on AWS, it's cheaper to buy a 60 grand FPGA engine than running a machine that's... Yeah,
I guess if you're doing production scale stuff on the cloud, I can imagine that does start to get expensive. Ours is kind of, I guess, more bursty, I guess. So it's not production scale. Like if I was in a sequencing institute and we were constantly running stuff using AWS, and I'm imagining, like a service center, like pushing out genome data, I imagine that's going to be really expensive. But for us, it's not constantly being used.
Are you planning for that in the future?
No, because we don't generate sequence data. We sort of pay someone to generate sequence data and then use the sequence data. We're more about the algorithm development than the analysis side.
Data science question. So which model do you use for the tribe? I'm curious because it could be...
It's not a machine learning model.
Yeah.
So Tribes is a pipeline that we built using existing tools, actually. And the crux of it is this, like I said, identifying these segments that are shared between individuals, and it uses sort of a dictionary approach. So it builds a dictionary of everyone's genome, and then looks for, using that dictionary, will identify regions that are matching between individuals. So Germline's the tool if you'd like to read more about the algorithm. That's the algorithm to be used within Tribes.
questions? Well, thank you very much, Natalie. Excellent talk.
Hi, I'm Greg Perl from the Onset Group. I head up the data science and analytic area there. I hope you enjoyed tonight's presentation. Always fantastic information that we get from that. The Onset Group are the premium technical recruiters across obviously data science, machine learning, NLP, artificial intelligence. I'm here to help organisations build their analytics team and their analytics function with superior talent. There's lots of people out there, really good individuals across a range of different sectors that could be beneficial. And obviously I'm also here for the data science community to assist with understanding the market, understanding what jobs are out there, how to get into data science. So please feel free to reach out. So again, it's Greg Perl at the Onset Group, and I look forward to seeing you at the next meetup.